We're going to go ahead and get started. Thank you all for coming today. I'm very happy to be able to introduce my friend and colleague, Dr. Matt Schuler. Matt is a community ecologist. He focuses mainly on how communities respond to anthropogenic changes. Currently, he's a professor at Montclair State University. Before that, he completed his undergrad in wildlife management from University of Wisconsin Stevens Point, his master's from Indiana State in thermal physiology, his PhD in ecology and evolution from Washington University in St. Louis. He did a postdoc at Rensselaer Polytechnic Institute, and now he's at Montclair. So Matt's been to a bunch of different places, and we're lucky to have him here with us today at Cary. So let's all welcome Matt.
Thank you, Max, and thank you everybody for coming. I appreciate it, and it's nice to talk to this many ecologists all at once and people interested in ecosystems. I'm in a biology department at Montclair, but we only have a few ecologists. I'm also the director of a doctoral program in environmental science, so I've reached out to quite a few people, and that's been a new opportunity of growth. And hopefully the things that I talk about today will lead to maybe some ideas and collaborations. I'm happy to talk about ways that any data I have or questions I'm asking could benefit you in any way, and any collaborations. Just feel free to email me if we don't have a chance to meet later today. Before I get started, I do have to actually give a huge thanks to my lab group, which has been supportive. I started at Montclair in 2019, fall 2019. I had one semester, my lab was under construction, so I didn't actually get started. January 2020, I was ready to go, I was all excited, and then COVID hit and crashed everything. But my lab has been there for me over the years, and I've had excellent grad students and support from undergraduates and the department and the college. I have to give a huge shout out to Dean Bobo, who is my partner, but also is a research associate and an idea center and somebody for me to talk with in terms of science. He does a lot of the bioinformatics and the data science with me, and has got me interested in microbial ecology. I steered away from it for so long, but he's pushed me into it. I'll talk a little bit about the research done by my PhD candidate, Arif Ahmed, who looks at multiple stressors. Also, Marissa Contreras, who's a new master's student in my lab looking at evolved tolerance of zooplankton to salinity, along with Nick Jacob, who is a master's student in my lab who's studying coastal lakes compared to inland lakes and looking at seasonal dynamics in zooplankton populations, but their projects are sort of shared in ideas. I'll talk about work done with Megan Klutz, who was a master's student in my lab, who recently graduated and is now a PhD student at the University of Maine. And I inspired her so much in aquatic ecology that she now studies bat acoustic research. So not everybody stays in aquatics, which is perfectly fine. I'll talk a little bit about Justin Howell's work of water quality, who's now a water quality specialist at NJDEP, and Kevin Petteroy, who's now with the New York DEP as in the Marine Sciences Department. And I'll focus a lot on work that Jared Vigil did as an undergraduate in the lab. We were working on projects, and he was testing a system to see if it would work for me. So he really helped me move out of the field and into the lab system, which was important because I was struggling to get field sites to work in New Jersey. A lot of people don't want you on their property asking questions about nutrient dynamics or contaminants, so you have to take things and bring them back to the lab, and he was really insightful in helping me do that. So I study urban landscapes and ponds, and I haven't always done that. I grew up in northern Wisconsin, so moving to New Jersey was quite a shock for me. It was really urban, very densely populated, and these are some photos that I took of some of my research sites, and very channelized rivers and birds on tires and garbage everywhere, and thinking about how can I ask my questions in this really challenged environment. There's lots of impervious surfaces, roads, parking lots. So what's interesting is this stream continues to flow, and it flows right under this parking lot and then disappears forever into a tunnel of unknown channels and eventually flows out into the Hackensack and into the ocean somewhere. But New Jersey has really modified their landscape, and it got me thinking, how does this loss of ecosystem structure and function affect the communities that live there, my background led me to study zooplankton, but since then I've expanded out to other systems, and I've really enjoyed thinking about how they're connected with zooplankton populations. Of course, zooplankton are important because they're sort of this intermediate negotiating the food web of energy flowing up to larger systems, but also they're important grazers in keeping algae populations down. And one thing that immediately happened once I moved to New Jersey is I noticed all this destruction, but everything kept flooding. It was constant flooding, and every time I would try to drive to work, there was a new flood somewhere. Oh, you can't drive there, that road's closed. And it's costly, it's damaging to people's lives, but it's also damaging to the ecosystem. And this is just an example of Atlantic City, which has seen a huge increase in the number of days that that city is flooded. If you ever, Atlantic City is known for casinos and gambling and its nice boardwalk, but a lot of time it's underwater. So you go there, you think you're going to have a nice vacation on the beach, but that's usually, the ocean comes to you pretty quickly. So I was studying this system of ponds and pools and vernal pools in New Jersey, and it flooded. And this was, in this particular case, this was one of the first times it had flooded in a while. I was talking to the homeowners in the area. They were very concerned, but I realized there was an opportunity to ask questions about how flooding might affect these urban wetlands. They're a rare ecosystem in New Jersey in the area where I live. If you go west, they're all over the place, or if you go south, they're all over the place. But where I live, they're relatively rare. And the one system that I'll talk a lot about today is called Walker Avenue Wetlands. I actually discovered it when, during the pandemic, all the parks were closed. New Jersey closed down and you weren't allowed to go anywhere. This dumpy little wetland next to where I lived in Wayne, New Jersey, was available. I could go there because nobody cared about it. It was a place where people dumped their garbage. It was a place where ATVs would go illegally and tear up ponds and go through the backwoods. So nobody cared that I was in there with scientific equipment. It was perfectly fine. I was birding, I was walking around and doing things. But it has a primary wetland. So a primary pond here, you can sort of see this large area. It's an artificial pond that was dug. When they made Interstate 80, they went through a bunch of wetlands, and this was a remediation project to try to mitigate some of the effects they had. But there's all of these vernal pools that lined up along this path around the outside of the pond. And there's a berm that's about 6 feet that separates the wetland, the main pond, from those vernal pools. And then nearby, there's the Pompton River. So it's a unique ecosystem, and it was not intended to flood. I don't think they build things intending to flood, but it was built to mitigate floods. It was supposed to help the homeowners in the area not deal with so much flooding, but it hasn't worked, and it's caused a lot of strife with the community. They're angry that they built this. They think that it's the cause of the floods, but it's really not. So I used an opportunity to study zooplankton and other community dynamics. Lots of species of amphibians and turtles here. I've got at least 8 species of turtle and I think 6 species of frogs, so there's good populations of those things. Of course, I'm interested in birds. As a personal passion, I like going out and birding and viewing things, so it's a really great hotspot for birding if you're into that. It's known as a migrant trap, so in the area, everybody knows about it, and it's one of those places you go if you want to find rare things in the area. But ecologically, there's also some really cool aquatic organisms there. So we got tardigrades in good numbers in a lot of these pools and in the main pond and wetland, and a good population of clam shrimp that grow in these vernal pools. So I've been looking at their population dynamics and studying them for a while. They're very sporadic, of course, as expected, but so we've been looking at their tolerances to salinity, and I'm not going to talk about that today, but if anybody is interested in clam shrimp biology, I'm happy to talk about that too. So after studying this for a very brief period during the pandemic, we were hit with 2 tropical storms right after one another. And Hurricane Ida and Tropical Storm Henri came through and dropped about 10 inches of rain each in the area, immediately flooding the landscape. And then there was another flood later that year, and then a flood in the spring of that year. So the timeline of this, I have my pre-flood sampling. I had just started going out there. I was really excited about looking at vernal pools. And then, of course, this flooding happened. So I have post-flood sampling in September and October. And then there was another flooding event, and I'll talk a little bit about some of the data from that sampling in December. It doesn't freeze that often in New Jersey anymore, so we still get samples in late December, which is pretty interesting. I have a lot of other samples that I took while I was out there. I was going out pretty regularly, and after some of the floods, I collected samples every 24 hours from all of the 20 ponds. Unfortunately, I have been struggling to find students who were interested in these projects and funding for some of the sequencing, so I still have a lot of the samples sitting around. I extracted the DNA. I've just been waiting for somebody to become interested in it, and maybe one day I'll find the funds to sequence those. But for right now, I just have a subset of samples that I'll talk about. What we did is we went out, and this is after the flooding. You can kind of see it's deeper than it looks. It's about 8 inches deep, but in the middle. And so these are very shallow pools. And we collected water samples for eDNA. We did shotgun sequencing from that. And then we looked at zooplankton, which were identified by microscope. We took water chemistry samples, which I used the Hawk spectrophotometer to analyze. And I learned some lessons there. That was a great, you should have done this after the fact. Hindsight is 2020. But the primary question I was interested in is how resilient are these ecological communities to flooding events? Do they actually change after it? Do you lose biodiversity, or do they bounce back pretty regularly? I expected that they would bounce back pretty regularly. This is a system that should be used to that, but I was curious what would happen, especially with the zooplankton, which were quite diverse in this area. I was getting 80 or 90 species of zooplankton throughout these vernal pools, which was pretty unexpected. And so I'm also thinking about the community, the composition of the species and sort of the state space, thinking about how they change over time. How long does it actually take them to bounce back? What other factors contribute to their response to flooding? And are there any other changes that happen in this ecosystem, nutrient dynamics or microbial dynamics, that might actually affect the rate of change? And so those are the questions we've been thinking about since these flooding events. Just some basic expectations. Flooding homogenizes the abiotic and biotic components of these communities, so we reduce the heterogeneity. And initially, these ponds were quite heterogeneous. I'll show you some of the data. The spread of just even phosphorus and nitrogen, there was a big difference in the pools, even though they're only 100 meters apart and they share the same soil and it's an artificial wetland. So they really should have been similar, but over time, they have diverged quite a bit. We expected that microbial composition would return to pre-flood conditions much faster than zooplankton. I think that's fairly obvious. They respond very quickly. We thought their functions and their composition would return relatively quickly, and zooplankton would take a longer time to respond, but and the abiotic conditions would follow whatever or correspond with the microbial conditions. So let's start with the abiotics. Just some simple things that we looked at a few other things other than this, but some primary data that we looked at. The first thing I was interested in, because I study salinization of freshwater systems, chloride increase, it spiked in these pools relatively quickly. So we have our August data. You can see that there's very little chloride in these systems. There's no road salt applied to this. There's no other source of anthropogenic salt. So these are sort of our baseline conditions that we would expect in a natural system in New Jersey. Anywhere from 1 milligram per liter to 4 or 5, maybe 8 milligrams per liter in the highest pool. And then it quickly jumps to over 20, and in some cases we had samples that were well over, in other sampling periods, well over 90 milligrams per liter. That did recover relatively quickly, but still remained more salinized than before. And to this day, after those floods, it washed in a lot of salt from nearby communities, and they have never recovered after it's been 5 years. And they've never recovered to those pre-flood conditions, those two big flooding events. Total phosphorus generally decreased on average. There's a lot of phosphorus loading in this community. It's an urbanized forest, so probably fertilizers and other things and allochthonous inputs into these pools, leading to high phosphorus concentrations. This is milligrams per liter, not micrograms per liter, so it is relatively high. There's also another problem that I found out about recently, the city of Little Falls, New Jersey has a lead problem in their pipes, as many communities do. And what they've done is they added phosphate to the water to sort of build this microbial mat around the pipe so that it doesn't interact with the lead. Great, I don't want lead in my water, so this is probably the best way without replacing the pipes. The problem is that about 25% of water going through the pipes leaks out and gets into the ecosystem. The amount of phosphate that they're adding is up to 2,000 micrograms per liter. So when you think about how much water might be leaking out of these systems and people watering their lawns and people filling pools or doing other things and then dumping that water into the system, it's actually adding a lot of phosphorus in this area where you wouldn't expect a lot of phosphorus. Total nitrogen, so this is what I was talking about with the heterogeneity of these pools. You can see the spread of total nitrogen in these systems. Some pools have very little, some pools have quite a bit. After the flooding event in September, you can see that it was homogenized, and on average it increased the amount of nitrogen. And again, by October, there was a huge spike in nitrogen. I'll talk about that in a second. If we look at this in sort of state space, we start here in this red circle in the lower left. You can see that there's a wide spread of abiotic conditions. These are NMDS plots, so this is NMDS1, NMDS2, and you can see that there's a good amount of variation after the flood. So this is October in the blue space up here. They're quite diverged in terms of their abiotic, their nutrients, phosphorus, nitrogen, chloride, and other things that we measured. And each time they flood, they sort of center right here. They become much more homogenized. So in this case, we have a first flood, and then this is a second flood that happened in November, and we measured after that. And sort of they sort of moved to this system. The nutrients never did recover to what they were before. And that's probably because there were allochthonous inputs, leaves, maybe fertilizer from nearby communities. I don't know. I'll talk about the problems with the way that I did the nutrients in a second. So looking at the microbial dynamics, and this again was, I'm not a microbial person, so this was my partner pushing me to try new things after COVID and move into new space, because I'm a zooplankton person. So we looked at family diversity of bacteria and archaea, and we see our pre-flood, there was a huge drop in diversity after the flood, and they never really did recover in terms of diversity. So we were interested in why that might be. We've investigated a little bit afterward, but the thing that happened is the actual composition did go back to what we had. So this is, again, our pre-flood. You can see in the red, it's behind a lot of other circles, but the green is immediately after the flood. So you can see this huge spread in diverse microbes that happen where our community increased quite a bit. The composition of species increased quite a bit, and each of the pools became very, very different, feeding on different materials at different rates, changing the dynamics. And so we thought, well, if that's happening, then we would expect that the ponds, the abiotic components would then diverge, and they would sort of go back to that heterogeneous state, this heterogeneous meta-community that happened afterward. But it didn't. That didn't match up. And so in October, the blue, it really did go back to the composition that was there before in terms of just the taxa. Looking at this in a different way, so we have our phylum relative abundance, and so the top is the pre-flood condition, and it's largely Pseudomonadata and a few other bacterial species, very little cyanobacteria in this system. And then we look after the flood, and there's huge spikes in cyanobacteria. And we were curious about what might be driving these spikes in cyanobacteria, because they disappeared after a couple of months. The cyanobacteria dropped off, and you could actively see it. You could see that there were these, the pools just turned this bright green, and there was lots of different taxa in there of cyanobacteria. And this is just looking at another way. So we have the percent of cyanobacteria reads out of all the sequencing reads that we got, and you can see that pre-flood and post-flood, there was a huge spike, and then 1 month after, they disappeared. So we've been thinking about what causes the cyanobacteria, and one of the thoughts that I had, I studied salinization of freshwater systems as a postdoc. Everybody was saying that chloride and salt pollution will drive cyanobacteria blooms because it will kill off the freshwater green algae or diatoms or whatever else is in these systems, and it will leave all the nutrient availability for these cyanobacteria that will take over the system, and it might produce cyanotoxins, and that sort of drives that system. So we're thinking, oh, it's probably the salt. We did see this huge spike in salinity. I'll come back to that in a second. And so with the microbes, we were also interested in functional diversity changes, and we did notice a few things that were interesting, but nothing that was really surprising. It wasn't like shocking news to learn about this, and some of this has led to new research that we're looking at. So on the left, the first thing that we noticed was that there's a spike in the metabolism, the genes that are regulating metabolism of aromatic compounds. We also have an increase in the metabolism of nitrogenous compounds, and then we have a decrease in the metabolism of proteins. So with the aromatic compounds, the runoff contains polycyclic aromatic hydrocarbons. So when you drive over roads, there's constant exhaust and carbons being put out on the road. Those then wash off into this pool, and that offers a new resource of carbon for a lot of these microbes, and they love it. They love taking advantage of that system. It also washes in a lot of leaves and organic material that they can take advantage of. So humic acid and tannins and other things that maybe were lacking in the pools before, they get a new resource for that. So it really did drive that system. And a collaborator and I, Alexis Kursagar, are looking at, she's really interested in PAHs and how they affect communities, so going from zooplankton to fish and how they might affect food webs. So we're doing some new experiments looking at PAHs and salinity and looking at food web dynamics. So hopefully more on that soon. The nitrogen metabolism is unlikely due to fertilizers. There's not any agriculture in the area. It's very urbanized, and there's very few lawns, at least very few nice looking lawns in this area. If you cross Wayne into a different region, maybe that would be more expected. There's a lot more fertilized lawns in that area, but for this, I don't actually think it was fertilizer. Also, because of the delay in the increase, I think that it was likely due to the breakdown of leaf litter and other allogenous inputs, so that new plant material, and that released organic nitrogen into the system. And this gets to part of the problem that I didn't ask the right questions at the right time because I didn't understand the system. It's part of the problem with moving to a new area and asking questions. You don't measure everything that you should have. And so I probably missed out on some, if I had broken down the nitrogen components into inorganic nitrogen, organic nitrogen, and ammonium and other things like that, I would have a better understanding of what exactly happened. But I can do that in the future, which is good. So we need to track that better to understand how microbial functions changed, and we can get better corresponding data. The protein metabolism is likely just a shift in the energetic costs. So it's more costly for the microbes to do protease catabolism, and so they probably just switched to a much easier and less costly system of breaking down different forms of nitrogen that came in with the flood. So that wasn't really too unexpected. But we were surprised that we didn't see bigger shifts in the functions of these communities, and why we expected that increased salinity would drive microbial functional diversity to do different things, and it really didn't. We didn't see massive shifts in functional diversity, which was kind of surprising. So flooding seems to have an effect, but not as big of an effect as we would have expected. Next, if we get into the zooplankton, so this is again work that was led by my grad student, Kevin. So if you're wondering why some of the graphs look different, it's because regardless of how much I work with the students and get them to do things in R, everybody has their own way of doing things in R, and this is what he really liked for his figures, so that's what we went with. But if we look at species diversity on the y-axis and we have our pre-flood condition, it really didn't change that much. Diversity of zooplankton did not change. We had about the same number of species. The abundances were about the same. It's just that the actual taxa changed. So the composition shifted quite a bit. So here in the blue, we have our pre-flood condition. September, we have this green, which is a little bit difficult to see, but we actually had a huge shift in zooplankton going from primarily rotifer-dominated communities with some cladocerans, smaller cladocerans like Chidoris, and then we shifted completely to Bosmina and copepod-driven systems. The rotifers were almost excluded after the flood, which was pretty interesting. And bosmina are not a small pond system, so I was very confused to see that many bosmina in there, and they persisted. The bosmina lasted quite a while in the system without too much trouble, and one of the questions I asked after this with another undergraduate student was, were there interesting changes in the egg bank, so the soil of each of these pools? And the answer is no. So the bosmina moved into the system. They did not actually sexually reproduce. They just died after a while. So they persisted for a long period of time. But when we tested the soil, we got almost the exact same species of rotifers and chidorids and some daphniids that we had before the flood. So even though this was a massive flooding event, and it shifted things, we didn't see huge shifts in the microbial functional diversity, and in the zooplankton community did actually revert back relatively quickly, and it didn't change the soil dynamics, so there's no change in the egg bank and no long-term changes in zooplankton in these systems. So zooplankton diversity was highly variable and remained unchanged, and composition shifted, did not return to pre-flood state that year, but it did in the next spring. So it only took one season to come back to that pre-flood state. Microbial composition was similar to pre-flood conditions. We did see this spike in cyanobacteria, and I'm gonna come back to that in just a second because we were pretty interested if that was driven by chloride. And the abiotic conditions did not return to pre-flood conditions, and that has persisted. So the nitrogen and phosphorus did not return to what they were before. Chloride never returned to what it was before. And part of the problem is I don't know if it's because of the flood or because people keep going in there and driving their ATVs around and mixing things up and making it difficult for me to study. So I've sort of left the system a little bit because after COVID, I realized that it was a good place to go birding. They realized it was a good place to do other things. And so it's been a little complicated going back in there. So the salt deposition that happened with the flood, water level increased by 3 meters after each of these storms. And we saw this spike in chloride. And we measured, so we looked at, we just used GIS and figured out elevational layers in this area. And we saw that the chloride concentration increased from the flood, I was supposed to say from the flood, and we estimated that the two storms deposited 150,000 to 200,000 kilograms of salt in this wetland area. It's a relatively large area, but that's still a lot of salt. And that was primarily one of the reasons we thought, okay, this must be something happening with the cyanobacteria and salinity, and we see this reduction in green algae or diatoms or other eukaryotic organisms. So that led to our next set of questions. Calcium and magnesium did not change. So we did measure calcium and magnesium over the study period. They actually didn't change. So this was primarily driven by road salt, sodium chloride, which is the main road salt that's used in New Jersey and in New York. Although we are shifting a little bit more to brines and magnesium and calcium-based systems, it's not in the town of Wayne. So our next question was, what did salt deposition, what effects did it have on the wetland community? There are lots of sources of salinity in freshwater systems. You have mining, agriculture, urban runoff, wastewater, and of course, road salt application. And it's really the over application or the misuse of these systems that leads to the increased salinity. If you're using these relatively rarely, they're not as bad. But salts enter freshwater systems very easily, so I always get frustrated with these open salt piles that are near water bodies, and I just watch like tons and tons of salt flow in every time that it rains in the spring. And so you get these huge runoff events, and you can see the salt enter freshwater systems. And this is not just a New York, New Jersey problem. This is a global problem. So this is a paper from Sujay Kaushal in 2021 looking at different aquatic systems all over the world. So Europe, Asia, South America, it's happening everywhere. And you have this increase in salinity. Like I said, we had a hard time studying things in the field, so we brought them into the lab, and Jared was really instrumental in understanding these microcosms and if they could be used for zooplankton research. I didn't want to bring adults and put them in because that's quite a shock going from a nice little pond community to a glass jar, but we took the soil out of each of the ponds and we used that instead. They have the Ephipia, the resting egg stages for each of them. And the idea was that we could get them to emerge in ideal lab conditions. I knew that there's resurrection studies that have been done on certain zooplankton species, but I didn't know that this would work for a large community study. And it turns out it does. We got pretty good data. So initially we compared the ponds that we have, the pools that we have on site, and the ones in the jars, and we got similar species diversity and composition in each of them. There were a few species that really don't like it. So copepods are not fans of glass jars, and they take too long to grow into adults. But they still do, it's just not at the same level of abundance. So we had the soil with eggs and resting stage bacteria and other things and phytoplankton, and we looked at their emergence in salty environments. I'm gonna show a series of plots where the control, so that's where we didn't add any salt, we just used salinity that was relatively similar to what we initially saw in the pre-flood conditions, about 5 to 10 milligrams per liter of chloride. And then we added different kinds of salts. So we have sodium chloride in purple, and you can see that we go from 50 milligrams, 100 milligrams, 200 milligrams, and 400 milligrams of chloride added to the system. So total dissolved solids is measured here on the y-axis. It's a proxy for the amount of chloride in the system. And then, so chloride concentration is on the x-axis. Here we have magnesium chloride in the middle and calcium chloride. So this is just to show you that I can add salt to water and I can get it to dissolve and I can put it in jars. So it's just an indication that it does actually work and these were saltier, and to orient you to the graphs. One of the things that we did not see was a reduction in zooplankton diversity. So salt, even at 400 milligrams per liter, we didn't see a reduction in zooplankton diversity or species richness. All that we saw was a reduction in abundance. And that was really only in the magnesium chloride and the calcium chloride. My previous research had showed that these are much more toxic to zooplankton than sodium chloride is. And it seems like in this system, there are other factors that relate to the toxicity of chloride. So the hardness of the water, for example, will determine how toxic this is. But we were very surprised that we did not get a huge reduction in abundance with sodium chloride, even at 400 milligrams per liter, because in previous experiments, 250 was about the limit that a lot of zooplankton could tolerate. But calcium and magnesium chloride were much more toxic. And we didn't see any effects on chlorophyll. So our green algae did not actually seem to change. And when we did some cell counts, it seemed to match what we were looking at. There wasn't a big reduction in the green algae of this system. That was kind of a shock to us. We expected to see huge reductions, especially with magnesium and calcium chloride. But actually in magnesium chloride, and I guess this is known in systems that if you add a little bit of magnesium, it's often limiting in systems, and the chlorophyll love it. They take advantage of that and they grow a little bit, and some other organisms take advantage of that. Mosquitoes love a little bit of magnesium in their water, so do amphipods. So that wasn't too surprising that it increased relative to the control. But otherwise, compared to the control, the abundance, the proxy for green algae was pretty stable across the concentrations of salt. We expected the opposite result for cyanobacteria. We thought we would see spikes in cyanobacteria. Everybody was telling us that you'll see spikes in cyanobacteria, and I have a reason why I think this result occurs in other mesocosms and doesn't occur in ours. So I'll talk about that in a second, but it has to do with the way the experiment is set up, using adults versus using egg stages. So sodium chloride, magnesium chloride, and a little bit of a reduction in calcium chloride, but especially magnesium chloride was incredibly toxic to cyanobacteria, and we saw huge reductions relative to the control. So our idea that salt pollution might have driven this spike in cyanobacteria doesn't seem to be holding up with what we see in a lab setting. We also noticed that there was reductions in turbidity and dissolved organic matter that we measured in the system. So we were able to, I don't have the graph for turbidity or for dissolved organic matter, but it matches effectively what the turbidity was. They're very close. Reductions from sodium chloride, magnesium chloride, and calcium chloride. They've all reduced dissolved organic matter and turbidity in these systems. And we noticed that there was a strong association between dissolved organic matter and the populations of cyanobacteria, the relative abundance of cyanobacteria. And Now what we're thinking is actually salt pollution might reduce turbidity in CDOM systems through flocculation and precipitation of these organic matters. Basically, like it just starts attracting these molecules together and they start sticking and they sink to the bottom. That becomes a problem for the cyanobacteria because they possibly need those sites to form their heterocysts in order to do nitrogen fixation. So that's our working hypothesis right now. Oh, I guess I do have CDOM. So you can see that CDOM was actually greatly reduced by the influx of the increased salt, more so in the magnesium chloride and calcium chloride. So they seem to cause this flocculation more than the sodium chloride. And I think this speaks to the danger of using alternative salts without understanding their potential consequences in these ecosystems. Sodium chloride, I mean, we all know that sodium chloride has its consequences and it's bad for systems, especially at high concentrations. But switching to other things because they're maybe safer or marketed as safer isn't necessarily the best option. And I've done research on the past on additives with salt, and if I have time, I'll talk about it for just a minute, like beet juice and distillation byproducts and brines that they add, and there's rich organic material, and they basically become fertilizers for these aquatic systems where they increase the algal growth, and that increases mosquito growth and all kinds of other problems that occur. And then you still have the problem of the chloride and the salt, and it's toxic if it has magnesium or calcium chloride in it. So sodium chloride is a problem, but maybe it's the least of our problems if we don't understand all the other consequences. The biggest thing that we can do, of course, is less salt, like just apply less and drive slower. But I live in New Jersey. That's hard to convince people of. And I'm partly to blame for that. I've gotten used to driving really quickly, and now I drive a BMW. So I've like really fit into the New Jersey mindset. And so now we're starting to look at the evolved tolerance of zooplankton in these systems because some previous research from a colleague of mine, Bill Hintz, and Kayla Coltsnow, and then my postdoc advisor, Rick, they looked at adults, they grew adults in different conditions and then looked at can they evolve rapid tolerance to these increasing salinities, and it turns out they can. So as adults, they have really rapid responses to increasing salinities in just a few generations when they're reproducing asexually. But I was curious about more constitutive long-term consequences of salinization. And so I switched to, again, thinking about this egg bank problem. Can we bring in soils with eggs in them from coastal environments where the zooplankton might have constitutive tolerance? They experience a lot of salt intrusion from seawater intrusion, storms, whatever might be occurring there. These are coastal lakes, so it happens a lot. There's just basically a beach separating them from the ocean, but they are freshwater. They have lower salinity than my tap water at my house, so it's still a freshwater system. And we can compare that to inland lakes that don't have that problem, but are getting salt pollution at different rates from either road salt or agriculture or some other type of pollutant. So Megan and Nick and Marissa have been looking at species diversity, so looking at the community. Does the community actually have this constitutive tolerance to salt based off of, so do they have natural salt intrusion? Are they close to the ocean? Or are they receiving anthropogenic salt pollution, road salt, whatever else it might be? And then we have the source salinity. So is it low, so it's very fresh water, or is it a little bit higher, maybe 1,000 milligrams of chloride per liter? Still fresh water technically, but it's on the higher end, and maybe that's just a natural source of a saline pond. And the idea is that if they are from high salinity in a natural salty environment where they're getting seawater intrusion, as we increase the salt, we would expect that diversity and richness and abundance would increase. So we have this increase. If we take it from a pond that has historically low salinity, so very fresh water, low salinity, as we increase salinity, we would see this opposite effect where they don't have constitutive evolved tolerance. But in the systems that are receiving anthropogenic inputs, we would just see a general decline. They have not yet evolved to tolerate increases in salinity. So we get reduced abundance, reduced diversity, reduced richness. Megan and I had grand plans. So we went out and we did this huge experiment. We collected samples from, I think, 6 or 7 different ponds, and we had different salt types. We had different concentrations. We had, I think, 6 different concentrations, and she wanted a bunch of replicates for each of her experimental units. And so my lab became filled with muddy jars, and we were really excited about it. But then reality hit that she would have to actually measure all of these things regularly. So we try to take abiotic measurements weekly. We try to take nutrients every other or every 3 weeks. And of course, at the end, she would have to count all of the zooplankton from this system. And so we ended up cutting out a lot of the samples because we didn't have the resources to actually do that big of an experiment. But the preliminary results do actually show that there might be, as we expected, this trend where the highest salinity increases. They seem to have this constitutive, community level constitutive tolerance to high salt, and the opposite. But then things just get weird. And so in the polluted system, they did both decline as we expected, but the high salt seemed to tolerate the low salt community much better than the low salt community did. We're not really sure what happened there. One of the problems is understanding the history, the context of each of these lakes or ponds is dependent on the data that are available about them. And when we think about New Jersey, we don't think about people being honest about what they've polluted. They try to hide it. It's a thing. It's what they have done in the past. And so there's a chance that we are thinking that this was a fresh, unpolluted pond, but there might be lead in the water. There might be cadmium or some other contaminant that we're not seeing. And that's a really common thing to run into in New Jersey. You don't know the history of these systems, and without testing everything, it's hard to say if there's some other factor that's changing the results so you don't find what you expect. So what we did was Nick was kind enough, he lives in Point Pleasant, New Jersey, and he was able to go out to a bunch of coastal lakes and increase the sample size and bring in more information into the system so that we could understand the zooplankton dynamics over time, measure the water quality. I'm working with Monmouth University, the Coastal Lakes Institute, to try to understand some of the abiotic components and the phytoplankton differences in these systems to add more data so we can actually get a true understanding of what's going on here. And if we look at so this is again Megan's experiment, if we look at chlorophyll, in the natural system, we don't see that salinity actually changes chlorophyll, which is pretty interesting, and we see this general decline in the polluted systems. So here we do actually see that salt reduced chlorophyll. And so again, it shows us that the type of answer that we get is context dependent. Maybe there might be some historical factor relating to that. And if we look at cyanobacteria, so it turns out the location, whether it was natural or anthropogenic, didn't matter for cyanobacteria. It just mattered if the source was low or high salinity. So if it was low salinity, we actually did see a decrease in cyanobacteria when we increased salt in the system, the blue line. So we see this reduction in cyanobacteria prevalence. And then in the high salinity system, cyanobacteria was perfectly happy. But once again, we did not see this spike in cyanobacteria that everybody has been telling us would happen. So I really do think that there's some other contextual factor affecting the cyanobacteria in these systems. And when we get cyanobacterial blooms, salt might be a contributing factor, but I think it's actually how it's affecting dissolved organic matter and turbidity of the system. And that has to do with how the salt washes in and then how it sits and then what it does in terms of flocculation and precipitation afterward. So high salt pollution reduces turbidity, and we don't see these strong effects on chlorophyll. High salt pollution does reduce cyanobacteria sometimes, but does not increase cyanobacteria. And so there might be that there's this reduced sites for heterocyst formation in cyanobacteria once you have salt pollution because your dissolved organic matter and turbidity is decreased. And high salt pollution reduces zooplankton abundance, but not necessarily diversity. Depends on the source, population, and history. And so I want to go back to the turbidity thing for just a second. The experiments that we did in the past, and what a lot of people have done, is they brought in adult communities. They go out and they collect a water sample, they filter it, and then they put that into the mesocosm, and then watch what happens. The problem is that those are already adult organisms, they might die immediately. So when you get a spike in cyanobacteria, it's because you've actually shocked the green algae and the diatoms and other things, and they've died. And the cyanobacteria are a little bit hardier, they reproduce faster, and so they probably just respond much better, and now you have all these released nutrients in the system, And so the answer that we've gotten from the past mesocosm experiments, these are mine included, I'm not blaming other people for this. I have many papers published with those results. But I think we were asking kind of an incorrect question, using an incorrect method to ask the question, what happens to cyanobacteria? And maybe some new results will shed light on that. And this is important because people are seeing cyanobacteria blooms in their lakes and they want to know why. And we often do know why, they just don't want those answers. But blaming road salt is a really easy thing to do, right? You didn't put it out there, somebody else put it out there, and now you want to blame them. So you're not going to blame the community for fertilizers on their lawns or agricultural systems nearby that are adding fertilizers. You're not going to blame those systems, you're going to blame the DOT drivers who are adding salt, even though there might not be that connection there, but it's a simple solution that they like to hear. So what can we do in this situation? Well, the first thing that we have to do in all systems is continue monitoring for salt pollution. If you don't know what the problem is or how far it extends, then you can't do anything about it. We've done a really great job with the monitoring, but we haven't increased the amount of ion-specific monitoring that we've done. And I'm guilty of this too. I bought all these wonderful probes when I first got to Montclair, chloride and sodium, and I have this Hawk spectrophotometer. I can test everything, magnesium and calcium. It's really wonderful. It's just very expensive and time consuming. And so we have to maybe consider, are there ways we can measure these things differently? Do research on experiments and understanding these systems, modeling, and then also just assessing a triple bottom line approach and thinking, in terms of society and the ecosystem and the economics, what can we do about this problem? And there are solutions. They might not be best for everybody, but that's usually what the triple bottom line indicates, is it's not best for everybody, but it's best overall. And triple bottom line isn't the only approach to this. There's lots of environmental modeling approaches to society and incorporating societal and business problems into it. And then of course, if needed, we can regulate systems. But regulations don't always work. They actually drive people away from what you're trying to get them to do. So we might just be better off managing the systems as best we can, thinking about changing how we use ions in the environment. Maybe we can actually make it a venture to make this a capitalist approach, right? We can say, oh, if you harvest the ions from this system, it improves our freshwater quality, and then you can use those ions for whatever it is that you're trying to do. And and the cost of desalinization is going down quite a bit as communities face this around the world, and so maybe we need to look into that a little bit more. And then of course we need to evaluate what we've done and see if it's working. So in New Jersey, one of the things that I've been focusing on is repairing habitat and repairing it to more natural state because it often reduces offload of wastes and pHs off of roads and helps communities. Reduce impervious surfaces, it's a big problem in New Jersey and it's a big problem in the Northeast, but we need to try to find ways to reduce them. And promoting native vegetation, a lot of people don't realize that invasive or non-native vegetation can actually increase the salinity of systems because they change the soil structure and they change the way that water flows over the landscape to basically flow right into the freshwater system, the surface water, instead of going into the ground where it might get into an ion exchange in soils. Reducing salt pollution. So instead of changing the type of salt that we use, actually think about how much do we use. And then, of course, reducing fertilizer use. And with the last couple of minutes, I want to talk about what I'm working on now. So I'm looking at studying microbial systems. I'm curious, what changes with ecosystem function, microbial function. And one of the things is we do know that in soils, salts do actually degrade microbial function. So it seems to be much more impactful in soil systems than it does in aquatic systems. But I wanted to know if additives like beet juice or other brines can actually improve microbial function by giving them some sort of carbon resource that they can use pretty quickly to recover from the salt pollution. But also I'm working with a chemist and she has created these artificial root exudates, so basically these synthetic chemicals in the lab that mimic chemicals that plants release, and we're trying to add those along with the salts in the soil. And so these are just some preliminary results. We do actually see that there's this spike when we add beet juice, or the same thing happens with the root exudates. We get this spike in microbial activity. But the problem is, these are plant growth parameters that we measured. So number germinated, plant height, number of leaves, and growth rate. And the biggest problem is that we see when we add high salt and beet juice, we do get a spike in microbial activity, but it kills the plants. So it actually incorporates a lot of fungi into the community. We get these huge spikes in fungal growth, not in bacterial diversity that we want to see in the soil, and it actually kills the seeds and they don't even germinate. So the opposite effect happened. We added the things that we thought the microbes would want, and it caused fungi to also want that, and then they took over and killed the plants. So we're working on that now, and we have a few experiments and field sites that we're studying, and I look forward to, if anybody has any ideas or ways that we can change our study system, I'd be happy to talk to you about that. So I'd like to thank my funding sources, and of course, again, I'd like to thank my lab group for all of the support, and thank you for all coming and listening to me talk. So I think what we're going to do is I'll take questions in here, and then if there's any questions online, and I'll repeat your question after. So if I don't repeat it, please remind me. Yeah, go ahead.
So I'm curious about the cyanobacteria responses and wondering about, I don't remember what happened to nitrogen and phosphorus with the flooding, but I'm curious if that interacts with, the need to fix nitrogen. And if that could, so if there's less nitrogen, then there's a need for more fixation.
Mm-hmm.
And so the cyanobacteria could win in competition. Or if there's more phosphorus, then that could also stimulate fixation. And then subsequently, what is the effects of the cyanobacteria on nitrogen levels, and potentially that could be stimulating the nitrogen metabolism?
Yeah, definitely. And we haven't looked at taxa-specific functions, which we probably should do. I think that's a really easy thing, not for me, for Dean to do, my partner. But in terms of, so nitrogen did increase. We had a spike of nitrogen metabolism, and we also had a reduction in phosphorus. And the reduction in phosphorus, even though it became lower, it's really thinking about the ratio of nitrogen to phosphorus, because the phosphorus is still really high. We're still at 1 milligram per liter in the pool, so it's still very abundant. But in terms of how it might have affected the system, we didn't, because there was a spike in nitrogen, we didn't expect the cyanobacteria to do so well, but they really did. And it might be that they're actually in there fixing the nitrogen. I guess we should break this down into individual components and see who's doing what. But yeah, that's actually a really good thought to who's contributing to this spike in nitrogen metabolism. Is it the cyanobacteria right after? Yeah, and then of course they died away relatively quickly. So we can look at when they're abundant and when they're absent and ask the question about nitrogen metabolism. Yeah. Yeah, thanks.
Really interesting talk. I was wondering if you could comment on what we might know about the mechanisms by which sodium, magnesium, and calcium affect the zooplankton communities, and if there are inferences that can be drawn about what we'd expect in terms of ability to develop tolerance to those mechanisms or things like that.
Yeah, so in terms of sodium, magnesium, and calcium affecting zooplankton, we know a little bit about their need physiologically, but they need really low concentrations of these things. So they don't actually need a large influx of this, and over 50 milligrams per liter of anything is excessive at that point. I know people have done work on the cellular mechanisms of algae, the food resources for the zooplankton, and found that they might actually have reduced food quality when you get too much sodium and magnesium and calcium. But otherwise, we've tried looking at zooplankton just in the lab and giving them magnesium and calcium, and as long as it's not associated with chloride, it doesn't seem to affect them as much. And again, the toxicity of chloride is really related to the other water parameters, so the hardness. And one of the problems is when you There's only a few ways to increase calcium and magnesium in systems. One is with chloride, and otherwise you're increasing the alkalinity of the system. And so it's like you're changing the dynamics of the system by adding something to test what it's doing. And so you're changing either pH or alkalinity, or you're changing some other aspect. So I don't know of anybody who's done it successfully, but I think it's an interesting question to ask: how much do they need physiologically, and where are their limits? Sort of thinking like a thermal performance curve or a physiological performance curve. You know, how much is too much? Yeah, Tara.
Thanks for the great talk, Matt. So I'm really curious, you showed that after the flood, there was a compositional shift in the zooplankton communities that kind of changed sequentially from October to November to December. And given that we've been thinking in our collaborative work about seasonal shifts in plankton community structure, I'm just wondering whether you have any kind of baseline data or expectations based on the seasonal fluctuations in those communities and the extent to which the post-flood communities matched or kind of deviated from those expectations.
Okay, so you're asking, sorry, I want to make sure I get this right. You're asking if I have any expectations for why the communities diverged away from their pre-flood state.
Yeah, we know that plankton community compositions are changing over the annual cycle.
Yes.
In lakes a lot.
Yes.
But we don't know much about what they're doing in ponds. And do you know what their kind of baseline seasonal shifts are like in your ponds?
Yes, okay, so the baseline, yeah, relative to a baseline, one of the things that happens a lot in ponds is during the summer, it's actually very similar to what you get in lakes. You get a huge increase in cladocerans in the summer. They tend to die off towards the end of summer. You get copepod communities that sort of persist throughout the season. And then in the fall, you get huge spikes in the number of rotifers. This system, rotifers seem to be the dominant taxa that's there. So we get like 30 or 40 species of rotifer, sometimes per little pond, which is really fascinating. And they persist for a long period of time. And then everything else blinks in and out really quickly. And so I showed you some picture of Daphnia that just exploded in a pond in a couple of weeks. I came back and they were gone. And that was in July, at where I would expect them to be there. But then they were loaded with parasites. And then they died out. And I haven't seen them back in that pond since. So the understanding the baseline conditions has been really challenging in the system. And again, that's part of the problem. I didn't know initially if it was because I was losing my mind thinking about what zooplankton should be doing, or if people are back there polluting the system. So some things that I didn't mention before. I came back into the site and there was somebody spraying pesticides. And I asked him, what are you doing? And he said, I work for the town of Wayne. This is my job to spray for mosquitoes. And I said, what are you spraying? And he said, I'm not going to tell you. You can file a Freedom of Information Act if you want to know that. And I was like, that's not making me feel good. And then, so in New Jersey, it's actually a law that they have to, before they protect a wetland from degradation, they have to protect the human population from mosquitoes. So they will spray for mosquitoes before they do anything else, which is really fascinating. Mosquito control is really big there. And mosquitoes are a big problem, and disease is a huge problem, so it does make sense. But the other thing they do is aerial spraying. So on top of him spraying each of my pools with some unknown pesticide, they do aerial spraying back in there. And so I realized that I might not be going crazy. I might just have a difficult time understanding this. And so that's part of the reason I've stopped studying this system is I can't understand the baseline of community dynamics that I would expect. Like I should get in the spring and early summer, these cladocerans, copepods persist throughout, and then I should get this spike of rotifers at the end. It does happen in ponds the same way it happens in lakes, but I don't always get it. So the year that I did this, I did get it. But that year they weren't spraying the pesticides and they weren't back there four-wheeling and doing other things. So yeah, it's after that I've been scratching my head trying to figure this out. But there's also other things like the parasites that I mentioned. So we had this huge spike in parasite communities, ectoparasites on the Daphnia. And up until talking to you, I was like, oh, that's somebody else's stuff, I don't know. And so I've realized that actually, we have some samples and that might have been driving some of these systems. So yeah, I would be interested in going back there and seeing if it was actually parasites and has very little to do. I did get the town of Wayne to tell me that it's a natural a pesticide only for mosquitoes, BD. So it's really just for them. It shouldn't affect any other invertebrate in the system. But there's lots of interesting things back there. I have never gotten a tick on me once in this system, and I've never seen one. And I have another friend who's doing dragonfly research, and we go out looking for mites on the dragonflies, and we've never found a mite on the dragonfly. So it's a weird system. There's lots going on there, and trying to figure out. And again, is it because it's urban? Is it because of things that people are putting in the system? Is it because of some historical contingency? I don't know. Or is it because of parasites? It's hard to say. Yeah.
All right, so we're at 12 o'clock. If you have any questions for Matt you didn't have a chance to ask, he'll be at lunch. He'll be here all day. But let's all give a hand for Matt.